Oke. Bismillahirrahmanirrahim. Assalamualaikum warahmatullahi wabarakatuh. Good afternoon, everyone. First of all, I would say thank you very much for all participants who are still attending this conference on the second day. Let me introduce myself. My name is Eka Prastia, and I will become a moderator in this parallel class with the topic of natural product and medicinal chemistry. And actually, in this section, we have 8 presenters who will present their papers. And I have divided in this class into 2 sections. For the first section, we listen from 4 presenters and continuing with discussion. And then following the second section, consisting of 4 presenters and also continue with discussion as well. And also important, if you have any questions, please kindly write your question on the chat box, or you can directly raise your hand via Zoom menu. And well, before we start the presentation, I will check the presenter that will present their article. For the first one, Ibu Andini. Yeah, I think.
Yes, I'm here.
Oh yeah, thank you. And then Mrs. Zetriana. Okay.
Present.
Okay, thank you. And Mrs. Melati Septianti. Okay.
Yes, present.
Okay, thank you. And Bu Risna. Oh yeah, Risna, thank you. And then for the second part, Mrs. Onisa Triwal Wafa. Yes. Is present here or absent?
Assalamualaikum warahmatullahi wabarakatuh.
Oh, waalaikumsalam. Okay, thank you. And then Mrs. Nadira Alifa. Nadira Alifa. Oh, still absent. Okay. And then Yasmin Eka Pratiwi.
Present.
Okay, thank you. And Ibu Galuh. Yes. Okay, thank you. Okay, everyone, we are going to the first presentation. Will be presented by Ibu Andini. She is a researcher at the Research Center for Chemistry. The title of the article is the synthesis of licarin A from isoquinol catalyzed by Brassica juncea peroxidase and its cytotoxicity on MCF strip 7 cell line. Okay, please for the committee, kindly play the video.
Assalamualaikum warahmatullahi wabarakatuh. Thank you for opportunity. Good afternoon, ladies and gentlemen. My name is Andini Sundawa from Research Center for Chemistry. Today I will present our paper.
Oh yeah, sorry, I think some kind of trouble from the video. Oh yeah, it's okay.
Assalamualaikum warahmatullahi wabarakatuh. Thank you for opportunity. Good afternoon, ladies and gentlemen. My name is Andini Sundawa from Research Center for Chemistry. Today I will present our paper entitled Synthesis of licarin A from isoquinol catalyzed by Brassica juncea peroxidase and its cytotoxicity on MCF7 cell line. The outline consists of introduction, materials and methods, results and discussion, and conclusion. As an introduction, according to the World Health Statistics reported by the International Agency for Research on Cancer, is still a leading cause of death worldwide. Comprehensive research in recent years has shown that regular consumption of vegetables and fruit may reduce the risk of contracting certain cancers. The phytochemicals derived from those vegetables and fruit are referred as chemopreventive agents, including genistein, resveratrol, curcumin, anethol, elagisic acid, isoagenol, and agenol. These chemopreventive agents have potential to be used as an adjunct to cancer therapy today. Isoagenol has proven immunity and the beneficial properties as antioxidative, anti-inflammatory, and anti-cancer. Isoagenol may also inhibit cell proliferation and induce apoptosis in human MCF7 breast cancer cells. Therefore, drug discovery efforts by exploring the potency and modifying isoagenol to develop novel and potent pharmaceuticals for breast cancer therapy are of considerable interest. Pepper described an enzyme of brassicodium peroxidase which catalyzes the dehydrogenative coupling of two isoagenol molecules in the presence of peroxidase. We designed dimerized isoagenol in order to improve their biological activity for anti-cancer activities. Here we use root enzyme from stem sawi hijau, Brassica juncea. A source of peroxidase was easily found and obtained since it widely distributed in nature belongs to the family of Horserades and also from the Indonesian native plants. For material and method are as follows. First, general experimental procedure. The fresh vegetable of Brassica juncea were purchased from local supermarket. Isoagenol was purchased from Sigma, and NMR spectra were acquired on Yol and ML spectrometer in Research Center for Chemistry. Gallin A was dissolved in PDCl3 containing tetramethylene as a chemical ship standard. And then extraction of peroxidase from Brassica juncea. Stem of Brassica juncea were washed, cut into small pieces, and then mechanically homogenized with sodium phosphate buffer pH 7 and blend still for 40 hours. The mixture were filtered and put in the refrigerator until used. The obtained supernatant contained Brassica juncea peroxidase as the main constituent. Next step, oxidative coupling of isoagenol catalyzed by Brassica juncea peroxidase. For Brassica juncea peroxidase in phosphate buffer, 200 ml was reacted with isoagenol, then add peroxidase. The mixture was vigorously stirred at room temperature for 50 minutes. After 15 minutes, the mixture was extracted with ethyl acetate for 10 times. The organic layer were combined and dried over natrium sulfate. Evaporation of the solvent yield the brown oil, which was purified by silica gel column chromatography with the ratio of solvent hexane ethyl acetate is 9 to 2. The oxidative coupling product was identified by this MS and NMR spectrometer. And then cytotoxicity assay on cancer cell line to evaluate cytotoxic effects of the test compound breast cancer cell line was used. It was cultured in DMEM supplement with 10% VBS and antibiotic. The first cell were placed in 96 well microplate at an initial cell density of approximately and culture in CO2 incubator. After 24 hours of incubation for cell attachment and growth, varying concentration of test samples were added, and the cells were incubated for another 24 hours. The formazan crystal was solubilized then by adding 100 microliter DMSO per well. And then absorbance of converted dye was measured at a wavelength of 540 nanometer by ELISA reader. Result and discussion. The oxidative coupling of isogenol catalyzed by crude Brassica juncea peroxidase produce of deisogenol. The product was obtained as a white powder. The Maya product was fully characterized by mass spectrometry and 1 and 2-dimensional NMR. The pure product was clearly identified by gas chromatography mass spectrometry with M-present value at 326 Dalton. In the spectrum, proton NMR indicate the presence of a specific spectrum of two aromatic rings and show the presence of two methoxy group. The other peaks show the presence of cis double bond of propane and propanil. The other signal indicated the presence of methyl and methane respectively. The structure was also supported by carbon NMR. Moreover, correlation proton and carbon NMR data by MQC experiment support also the structure elucidation. For complete functional groups, position was supported by HMBC experiment. Based on this NMR data and compare to Licarin A data from literature, it was identified as Licarin A. The result of cytotoxicity assay, those response curve for the effect of Licarin A and doxorubicin on MCF7 cell viability after incubation for 24 hours. The R quadrat values for the DLT of Licarin A and doxorubicin are 0.97 and 0.96 respectively. The IC50 values of Licarin A and doxorubicin are 49.95 program per milliliter and 0.88 microgram per milliliter respectively. In conclusion, Licarin A could be synthesized by dimerization of isogenol follow crystallization catalyzed by crude Brassica juncea peroxidase. Mass spectrometer and nuclear magnetic resonance NMR analysis describe the structure of the synthesized Licarin A. This compound show marginal cytotoxicity effect on NCF7 breast cancer cell compared to the positive control doxorubicin. This result indirectly verify our previously published in silico test that show if Licarin A was not a potent ligand for receptor AR alpha. These are the reference. That's all I can present. Thank you for attention. Wassalamualaikum warahmatullahi wabarakatuh.
Waalaikumsalam warahmatullahi wabarakatuh. Thank you very much for your kind presentation, Bu Andini. Okay, we're going to the next presenter that will presented by Mrs. Jetriana. She is a researcher at the Research Center for Chemistry, with the title of article is Insight Study of Trans-ethyl formetoxinamide Isolation and Hydrolysis. Okay, please the committee kindly play the video.
Bismillahirrahmanirrahim. Assalamualaikum warahmatullahi wabarakatuh. Good afternoon. Thank you very much for the time. First of all, please allow me to introduce myself. My name is Detriana Putri Tahrim from Research Center for Chemistry LIPI BRIN. I would like to present our study titled Insight Study of Trans-ethyl 4-methoxycinnamide Isolation and Hydrolysis. The presentation is divided according to this session. Trans-ethyl 4-methoxycinnamide or ethyl para-methoxycinnamide is one of the major compounds isolated from Campherina galanga, a native tropical plant that grows in Asia. This compound constituent responsible for anti-inflammatory effect in Campherina galanga extract and showed nematicidal activity and larvicidal activity. Due to the importances, trans-ethyl 4-methoxycinnamide isolation is extensively studied. The trends are utilizing ethanol for the isolation, which started in 2011 when Susanna's group utilized percolator. The following year of 2014 and 16, Omar et al. And Wu et al. Combined two techniques that involving the supercritical fluid extraction. The current study of Hakim in 2018 utilized supplementation with ethanol as a solvent. From the extraction efficiency in trans-ethyl 4-methoxycinnamide isolation from Campherina galanga calculated by the reported study in 2019, it is showed that the order for suitable solvent utilization is start from acetone, methanol, chloroform, dichloromethane, and then the least preferred of less polar solvent of hexane. The serial extraction conducted with supplementation of less polar solvent of petroleum ether, followed by chloroform, and then liquid-liquid separation using hexane, can obtain the pure crystal after recrystallization step. In spite of this complex extraction, in 2017, Komala et al. Extract trans-ethyl 4-methoxycinnamide by simply storage in refrigerator of hexane extract to form an easily to separated crystal without any further recrystallization. Based on the success of the previous study for the isolation of trans-ethyl 4-methoxycinnamide from Campherina galanga, thus, in the aim of this study is to revisit isolation of compound trans-ethyl 4-methoxycinnamide from Campherina galanga, and then utilizing less polar and easier to handle solvent of hexane at room temperature without any supplementation or percolator, and complete the structural elucidation by conducting the hydrolysis in alkaline condition. First, the Campherina galanga rhizome dried powder is underwent maceration, filtered, and the filtrate is concentrated under vacuum. From this step, there are two methods that conducted in isolation. Method A, which the crude extract was stored in refrigerator, and method B, which the crude extract was left sit at room temperature. From both methods, the yellowish crystal was obtained and can easily separate it from the oil. The pure trans-ethyl 4-methoxycinnamide from method A was obtained by several recrystallization with hexane ethanol 9-to-1. Meanwhile, the method B utilized flash column chromatography. From method A, 0.07% weight per weight of pure colorless crystal of trans-ethyl 4-methoxycinnamide, and as for the method B, the higher isolated yield, which 0.83% weight per weight of pure trans-ethyl 4-methoxycinnamide can be obtained. From this result, we conclude that the recrystallization method firstly introduced by our group, is needed more intense effort to isolate pure ethyl trans-ethyl 4-methoxycinnamide to avoid column chromatography. The pure isolated trans-ethyl 4-methoxycinnamide then subject into the hydrolysis by sodium hydroxide and utilizing two methods, the conventional stirring and also sonication. The excellent result was obtained from both condition and the sonication method, which firstly introduced in this study can replace the conventional stirring method. The successful of isolation can be shown in proton, carbon, and mass spectrum from pure trans-ethyl 4-methoxycinnamide and its hydrolyzed form of trans-4-methoxycinnamic acid. From this proton and carbon NMR, the absence of the ethyl ester counterpart of trans-ethyl 4-methoxycinnamide at the chemical shift of 4.25 and 1.33 ppm. And carbon NMR chemical shifts showed that 60.5 and 14.5 ppm indicates the successful of its hydrolysis into trans-4-methoxycinnamic acid. In line with this result, the loss of ethyl ester can also confirmed by the result of mass spectrophotometry. In conclusion, the isolation of trans-ethyl 4-methoxycinnamide can conducted by maceration with hexane at room temperature that needed intense recrystallization step and followed by hydrolysis of the isolated compound to trans-4-methoxycinnamic acid for completing the structural elucidation. The research was part of the penelitian program prioritas nasional Deputi Bidang Ilmu Pengetahuan Teknik Lembaga Ilmu Pengetahuan Indonesia tahun 2021. This is the end of my presentation. Thank you very much.
Yes, thank you very much, Mrs. Zetriana, for your clear and interesting presentation. Okay, everyone, we will going to next presenter. Will be presented by Mrs. Melati Septianti. She is a researcher at the Research Center for Chemistry, BRIN. And the title of this article, Formulation and Stability Study of Fermented Soybean Extract Nano-emul Gel. Okay, the committee please kindly play the video.
Assalamualaikum warahmatullahi wabarakatuh. My name is Melati Septianti from Research Center for Chemistry, National Research and Innovation Agency. In this opportunity, I would like to present our work. The title is Formulation and Stability Study of Fermented Soybean Extract Nano-emul Gel. This is the introduction. Skin is our part of body that exposed of excessive sunlight and UV light that trigger free radical formation which cause aging. To prevent this type of photoaging, we need the substance called antioxidant. It is the active agent, protects the skin by absorbing UV radiation, inhibiting free radical reaction, and regulating endogenous antioxidant systems. The natural antioxidant resources can be found in fermented soybean, which is rich of isoflavone. This extract will be formulated in nanoemulsion, which have advantages such as enhance the bioavailability of lipophilic compound by improving topical absorption capability, has better permeation properties and better therapeutic efficacy, less stickiness properties and better spreading characteristics, and higher stability against coalescence. Thus, the study aimed to enhance topical usage of fermented soybean in nano-emul gel formulation, which allow higher dissolution ease of active agent release, and more stable system. The methodology used in this study consists of 4 steps. First step is fermented soybean extraction. First, fermented soybean crushed using blender and then macerated in ethanol with ratio 1:2.5 for 7 days, and then evaporated to obtain viscose extract. The next step is nano-emulsion formulation. First, we mix oil phase that consists of avocado oil, PMO, and PDO as surfactant, and then mix water phase that consists of fermented soybean extract, glycerin, propylene glycol, nanosilver, oxyl, and water. And then oil phase was added into water phase under high energy homogenizer ultraturax with speed 12,000 rpm in 30 minutes. Next is nano-emulsion formulation. First, we mix water, propylene glycol, and carbomer at temperature 50 degrees Celsius. The nano-emulsion gradually added with ratio 1:10 under stirring, and then the triethanolamine was added under stirring. And the last is characterization with the parameter antioxidant activity, total phenolic content, total flavonoid content, and stability test that's comprised of physical appearance, droplet size, pH, viscosity, and spreadability. This is the formulation of fermented soybean extract nano-emulsion formulation, and then the gel preparation formulation. The variation that we done in this study is the ratio of the PMO and PDO as surfactant. This is the result of the phytochemistry characterization. The fermented soybean extract antioxidant activity has the value of AAI, relatively low antioxidant activity. The low antioxidant activity caused by the source of the fermented soybean from the free market with uncontrolled fermentation condition and allowed contamination from other microorganisms which limited performance of the process. The antioxidant activity could be enhanced under controlled fermentation and more sterile condition. In Sambron et al. Study, ethanolic fermented soybean extract with controlled fermentation condition using pure isolate produced strong antioxidant activity with IC50 29 ppm. Thus, this indicated a potential antioxidant resources from fermented soybean extract. As the result of PVC and TFC, it was reported that phenolic compounds contribute to the antioxidant activity in vegetable, fruits, and grains. The results showed that fermented soybean extract contains flavonoid content. It was studied that fermented soybean product rich in isoflavone glycans, such as diazane, glycine, and genistein, which also corresponded with radical scavenging activity. This is the result of stability of nano-emulsion formulation, which have done using the accelerated stability test, where the sample was stored in 5 degrees Celsius and then altered into the 40 degrees Celsius for every 24 hours. So this is the result that show the physical appearance of nano-emulsion showed semi-solid shape with no separation appeared in the system as seen on the picture. Carbomer as gelling agent form hydrogen bond when it mixed with water. To form gel system, triethanolamine was added, then ionized carbomer produced negative charge along polymer backbone that caused electrostatic repulsion. This repulsion configured three-dimensional structure, thus form semi-solid gel. The milky color characterized as emulsion system with droplet range from 200 until 500 nanometers, which had turbid appearance. The observation for 3 weeks showed that there were no significant change and showed good stability on nano-emulsion physical appearance parameter. This is the droplet size of nano-emulsion formulation. As we see in that there were slight increase over 3 weeks observation. This might be occurred because Oswald-Ripening phenomenon, wherein the small droplet size yield higher solubility, thus it form larger droplets. In week 3 of F2, smaller droplet size was characterized. This might happen because of the shear produced by particle-particle interaction during measurement that could affect scattered light intensity, thus generated small droplet size. However, F2 had the smallest average droplet size, if caused by the surfactant ratio on each formulation. The surfactant used in this formulation were PMO and PDO with the HLD value respectively 10, 12, and 9. High HLD value is suitable to form and stabilize oil and water emulsion, which is commonly ranged between 8 until 14. The hydrophilicity could encapsulate more fermented soybean extract, which was soluble in water, faced with smaller and even droplet size as performed by F2. F2 showed narrower distribution compared with F3, while F1 showed bimodal distribution as seen on the picture. In the study, PI of all formulations were relatively monodispersed with range 0.05 until 0.4, except F1 with PI 0.5. This is the pH value of the formulation. It is ranged in 6 until 7. Carbomer dispersed in water form acidic colloidal dispersion, and when neutralized it produce viscous gels with more stable viscosity in pH 6 until 11. The average of pH also similar with human skin pH, which is ranged in 4.5 until 6.5. Thus, this formulation can be applied on skin with minimum risk of irritation or erythema. However, there was a slight pH decrease over 3 weeks observation. This caused by the nanoemulgel contained surfactant PMO and PDO, which was unsaturated fatty acid, and emollient avocado oil, which was rich of monounsaturated fatty acids such as oleic and palmitoleic acid. The unsaturated fatty acid might hydrolyze during the stability test and produce carboxylic acid, which caused the pH decrease. This is the viscosity. The viscosity didn't change significantly and also showed relative good stability. Carbomer colloids, when it is added with TA, form three-dimensional structure with stable viscosity in the pH above 6. It corresponded with the stable pH that observed in the previous discussion, which result in an consistent viscosity. The selection PMO and PDO as surfactant also support the stability because the non-ionic surfactant was stable in acidic or base condition and suitable for semi-solid preparations such as cream, gel, and paste in oil-water type emulsion. The presence of the surfactant promoted laminar liquid crystal phase in a water oil emulsifier system, which increased emulsion stability by forming a layer around the emulsion droplets, conducting the van der Waals interaction between the droplets, increasing repulsion charge, and enhancing the viscosity of the nanoemulgel formulation. This is the spreadability of the formulation. It is ranged between 5 until 6 centimeter, and this value categorized as a semi-fluid cream with standard spreadability 5 until 7. The spreadability of the nanoemulgel was relatively stable with insignificant change during 3 weeks observation. The average spreadability of F3 was wider compared to F1 and F2. It corresponds with its viscosity as mentioned in the previous discussion, where the viscosity of F3 was lower compared to the viscosity of F1 and F2. The physical stability such as stability, adhesion, and spreadability immensely related to the viscosity of which lower viscosity resulted wider spreadability. So the conclusion of this study, the fermented soybean extracted by ethanol produced crude extract with yield 9.475%. The phytochemistry had been characterized by the value of IC50, PPC, and also TFC. The nano-emulgel formulation from fermented soybean extract show relative stable performance after accelerated stability test due to its nanosite emulsion. F2 showed the smallest and narrow droplet size distribution with average size 140 nanometer, EI 0.2, with suitable surfactant ratio PMO/PDO 2/1 by HLB 11. Fermented soybean extract formulated in nano-emulgel preparation could be a potential antioxidant resourcer for cosmetic when an is active agent release, pleasant texture, better spreading characteristic, and external stability. That's all the presentation for me. Thank you for your attention. Wassalamualaikum warahmatullahi wabarakatuh.
Waalaikumsalam. Thank you very much for your nice presentation, Mrs. Melati. Okay, everyone, we're going to next presenter that will be presented by Bu Krishna with the title of her article is Phytochemical Constituents and Bioactivities of Dewan Daru Fruit Extract. Okay, the committee can play the video.
Good afternoon, ladies and gentlemen. I am Krishna Triana Dewi from Research Center for Chemistry, National Research and Innovation Agency, BRIN. Today, I am going to present our paper titled Phytochemical Constituent and Bioactivity of Dewan Daru Fruit Extract. My outline presentation are introduction, experimental section, result and discussion, conclusion, and references. In my introduction, Dewan Daru fruit is belong to Myrtaceae family. A scientific name is Eugenia uniflora, and Eugenia uniflora has various local names such as Suriname cherry, Brazilian cherry in South America, or Pitanga in English, and Indonesia. This plant now as Dewan Daru or Tamarin Selong or Blimbing Londo. This plant native in South America countries, but in Indonesia we can find this plant from in the Java and Sumatra Island. Dewan Daru plant is rich in benefit. Dewan Daru is now as medicinal plant by the Brazilian people. They use the leaf and fruit of Dewan Daru as medicine for diarrhea, fever, hypertension, lowering blood cholesterol, obesity, anti-diabetic, and also for antihypertensive. And common essential oil from the leaf is found in many beauty cosmetic products. Not only for leaf and fruit, they also use seed for anti-diarrhea, diuretic, antifebrile, and also for anti-diabetic. More recently, scientists become more interested in Dewan Daru. However, Dewan Daru fruit is not widely known in Indonesia, and limited studies are related to the pharmacological property of the fruit extract. Therefore, this study, our objective, this study to conduct a comparative assessment of fresh and dry seed and pulp on the phytochemical profile and the biological activity, including free radical screening effect on DPPH, enzyme inhibitory activity, and also for antibacterial activity. Here are the experimental. The fruit is we collect from Lembang, Bandung in June 2020, and the taxonomy plan identification was confirmed by biologists in Research Center for Biology. First, fresh fruit is around 14 kilograms. We separated carefully for pulp and also for seed, and then fresh and dry seed and pulp we measured it with ethanol 98%, and then evaporate to obtain the dry extract. And step 2, we conduct the phytochemical analysis to identification a common metabolite secondary in the extract, such as terpenoids, alkaloid, flavonoid in standard method, and also we determine the total phenolic compound and total flavonoid compound. And for bioassay analysis, we use antioxidant against free radical scavenger effect, and then also for inhibitor reactivity, two enzyme alpha-glucosidase inhibit alpha-glucosidase from Saccharomyces cerevisiae, and pancreatic lipase enzyme for anti-obesity. And for antibacterial activity, we use positive gram-bacterial and negative gram-bacterial using this infusion method. The result we can see here for the yield and also for qualitative secondary metabolite, common secondary metabolite in the extract. We can see this table. We can see here that fresh pulp extract and dry pulp extract has different for yield percentage, because the fresh pulp extract is high content of the water, but in dry pulp extract is high to carbohydrate content. And drying also is increased for percentage of the yield from the seed extract. But for qualitative analysis of common metabolite secondary, drying is no significant effect for the result. We can see only for drying pulp extract here that dry pulp extract has a positive result for saponin, but in the fresh pulp extract is negative. Biological activity as antioxidant is common related to the flavonoid and phenolic compound in the extract. So we determine the total phenolic compound we express in gallic acid equivalent, and total flavonoid compound expressed in quercetin equivalent per gram extract. You can see here that both of the pulp extract, both of the fresh or dry, is low content for phenolic and flavonoid. But for seed extract, the drying seed extract higher than fruit fresh seed extract for total phenolic and total flavonoid content. Even in some previous report, like Chile in 2011, reported that Dewandaru fruit extract contain phenolic and flavonoid compounds such as cyanidin, myrcetin, quercetin, and also their glycoside is known to correlated with antioxidant activity. But in our study here, that the content of phenol and flavonoid in the pulp extract is low. And we determine for scavenger effect on TPPH free radical, that we can see here that the pulp extract for fresh or dry is no effect of the for TPPH free radical, but the opposite. On the other hand, the dry seed extract, if fresh or dry, has activity for as antioxidant or free radical scavenger effect, which the IC50 is around 50 and 20. So the dry seed extract is higher than a fresh seed extract. But for inhibition activity on enzyme alpha-glucosidase or pancreatic lipase, we can see here that pulp extract, both of the fresh and dry, has significant effect against this both the enzyme. So it's agree with the previous report that the fruit extract, but they use from fresh fruit has a significant value for IC50 against enzyme alpha-glucosidase, and also for pancreatic lipase with the IC50 is around 12 and 2 milligram per milli. And for antibacterial test, we can see here that pulp extract no significant effect to inhibit the E. coli, Staphylococcus aureus, and Bacillus subtilis. But for seed extract, dry seed extract, also the potential and zone of inhibition is around 10, 20, or 13 for each bacteria, but fresh seed extract is not active. Now, so inhibition is so that means the drying process of the seed is increase the antibacterial activity and also for antioxidant activity. And why this seed extract has the bacterial activity is maybe correlated with the sesquiterpene and terpene in the extract, because some reported report that the seed extract content of the essential oil and also found in the lip and is linked to the antibacterial activity. So from the research study here, we can conclude that the pulp extract have potent inhibit the alpha glucosidase and pancreatic enzyme, but the seed are more active as antioxidant and antibacterial. So the drying process is more increase the activity for a seed, but is decrease activity for pulp extracts, maybe damage or decrease the flavonoid or carotenoid content in that pulp extract. So the present study indicates that the pulp and seed of Dewandaru fruit have promising bioactivity for the food industry to develop as functional food or supplement. However, further investigation is needed related to their bioactive compound. And also, we still conduct analysis for LC-MS to investigate the diet bioactive compound in all extract. Here, the references we use in this paper, and we want to we analyze from Mr. Mara Toha from LIPI AD, who providing a sample for us, and also we thanks to organizer to provide me with an opportunity to present our paper, and thank you for audience for your attention to our presentation.
Okay, thank you very much for Bu Krishna for your nice presentation and interesting regarding the Dewandaru fruits. And okay, everyone, for the next session, we will have the discussion Q&A. Actually, we have maybe 50 minutes until 2 p.m. And I have already see in the chat box, some presenter already mentioned the questions. Okay, I will explain for the first question from Zetriana Prin to Mrs. Andini. The question is, for the pH of the reaction is just into 6 to 7. I would like to know whether the pH is the optimum condition for the reaction. Is there any approach in the study for other pH? Okay, Bu Andini can answer this.
Thank you. I'm sorry, before, may I answer in bahasa?
Oh yes, of course. Okay.
Okay, thank you, Betri. Jadi kondisi pH pada reaksi ini memang kita set di pH 6,7. pH tersebut merupakan memang dari hasil optimasi ya. Jadi sebelumnya memang kita optimasi beberapa pH dan yang paling bagus reaksinya stabil dan optimum pada pH sekitar 6 sampai 7. Mungkin demikian. Terima kasih.
Yes, that's the answer. How about the Mrs. Jadriana? Maybe they have the response or enough?
Very clear. Thank you very much.
Okay, thank you. Well, maybe actually I will confirm that Bu Andini using the crude enzyme, right? Crude enzyme. Oh yes.
Yes. Crude enzyme.
Crude enzyme.
Yes.
Okay, thank you, Bu Andini. For the next questions from Bu Sofa, Prin to Ms. Andini also. And the question is, why licarin A has lower activity than doxorubicin? Have you done another bioactivity of this compound? Thank you. Yes.
Baik, saya coba menjawab. Jadi kenapa likarin A ini aktivitasnya lebih rendah dibanding doxorubicin? Jadi sebelumnya publikasi sebelumnya memang dilakukan in silico terhadap senyawa likarin A ini, di mana in silico terhadap reseptor estrogen reseptor alfa. Jadi dari hasil in silico tersebut memang si likarin A ini binding affinity-nya lebih lemah dibandingkan dengan doxorubicin. Jadi kalau doxorubicin ini gugusnya aktif dan berikatan dengan reseptornya, sedangkan likarin A ini tidak berikatan dengan reseptor dari si estrogen alfa tersebut gitu. Jadi aktivitasnya untuk lebih rendah dibandingkan dengan si doxorubicin ini. Untuk bioaktivitas yang lain memang kami belum mencoba. Jadi baru mencoba untuk ke sel MCF7 ini, belum mencoba untuk ke sel breast cancer yang lainnya. Jadi memang itu hasil dari in silico sebelumnya. Mungkin itu Bu Sofa.
Yes, thank you, Bu Andini. Maybe Bu Sofa will have the response or is.
Okay, thank you very much for the answer. Thank you.
Okay, thank you. Okay, sorry. Okay, for the next question from Jetriana Prim to Ms. Melati. And the question is how to identify or quantify the smell of soybean odor after the formulation. Is there any study to remove the slightly soybean odor? Yes, Ms. Melati, time is yours.
Thank you for the question, Mrs. Veteriana. In the study, we didn't identify or quantify the smell of the soybean odor, and we are not removing the odor. But to modify the odor, we can add the fragrance into the formulation. Thank you.
Yes, thank you. Mrs. Jadriana?
So, Ms. Melati, how can you identify that after the formulation still a little bit smell of the soybean odor from phenolactic or another study you've been?
It's an organoleptic study. Yeah, we are not using instrument, but we use organoleptic study.
Okay.
Thank you.
Okay, thank you for Ms. Melati for your nice answer. And then the next questions from Mas Abdi, still to Ms. Melati. And the question is why you did not try to use other gelling agent for intense PMC or CMC in this nano emulsive gel? Yes.
Thank you for the question, Mr. Abdi. So we use carbomer as a thickening agent because it is stable and very stable in stabilizing the emulsion. And then it has a high viscosity, and also the important part is it is produce the clear gel and also a smooth texture, so like a smooth watery texture that is really comfortable for the cosmetic uses. So we also use HPMC2, but in a different application. So and the result of the formulation is quite a bit sticky, and we use it for different application, not for cream, nano emul gel, but for peeling mask. Yes, so it has a different texture compared with the carbomer.
Yes, Mas Abdi, maybe give the response.
Oh, yeah, clear. Maybe additional question, if possible. in this study, your focus on the stability, formulation, stability, and characterization, right? For the next, I do believe that you will you will want to know the bioavailability and activity as well. In your opinion, what kind of study that will you do for the next? If you want to know the bioavailability and the activity this formulation to be applied in patients, something like that.
It is yes, it is important to do a further study. So the next step after the formulation we usually do first is the I forgot, irritation test. Yeah, first we do the irritation test, and further we can do the efficacy test. It is for anti-aging, so we take the parameter of the radius of the, what can I say, the texture of the skin, something like that. Yet we cannot do it alone for the efficacy test. So we need to have a dermatologist assistance to do all of it. So maybe it needs a lot of time and a lot of cost. So we have to, let's say, plan it carefully. Thank you.
I think you're going to do the penetration test as well, right? So I didn't find any transdiffusion test, transdiffusion cell that will assist us to perform any bioavailability and efficacy of the topical application. I think maybe you can suggest to our management to buy transdiffusion test, transdiffusion cells, so can support us.
I have heard about that when I go to Sirim in Malaysia. They have maybe we can have it one day.
Thank you so much.
Okay, yes, thank you so much for the nice discussion and interesting discussion. And okay, for the next questions, I think it's the last questions from. Two more questions from Zetriana Prin to Risna. The question is there any pretreatment into the pulp? I would like to know whether you remove the peel in advance. Yes, Risna. Thanks.
No, we no pretreatment for separated pulp and seed because because the Dewandaru is like cherry fruit, and then the size also small, like jambu air kancing. So that's no need to separate peel and pulp. Okay, see, clear for your question, Mrs. Chetri?
Bu Risna, is there any possibilities that might be the peel can give the antioxidant activity rather than the fruit itself?
Because like cherry, it's so not so not just chicks pulp. So that's maybe it's difficult to separate it peel and then pulp and then seed. So you can some literature also said that the seed also can edible seed. So for Brazilian pork, they in diarrhea they consume all the fruit, the pitanga fruit seed, and also because it's just small. It's a little bit more smallest than cherry.
Thank you very much.
Okay, and for Dr. Ahmad, say that's because our study here that total phenolic and also total flavonoid is almost similar, but we can directly say that's maybe this the same compound, because for phenolic we use gallic acid as equivalent, and for quercetin is flavonoid. So we can see that different structure from gallic acid and also the flavonoid. So for we still continue to identify the metabolic compound in a sample by CMS, and then compare to the some references, but almost all references say that the compound in the Titanga or Dewan Daru is anthocyanin, because the color from the green to the purple. So the content is depend on the maturity level. So we can directly, for me, we can say that the phenolic and flavonoid is maybe the same, because we use a different compound to identify or to equal or to calculate the total phenol and also total flavonoid. Yes.
Okay, that's the answer.
Question,
yes.
Dr. Ahmad.
Okay, clear.
Thank you for your.
Okay. Yes, thank you, Pak Ahmad and Bu Risna, for I think it's the last discussion. Okay, everyone, we'll going to the second session of this parallel class, and in this section we will have 4 presenters. For the first one, the presenter will be presented by Onisa Triwalwafa. She is a researcher at the Research Center for Chemistry, with the title of its article, The Effect of Nanoparticles Dewan Daru Fruit Extract to Reduce Total Cholesterol and Hyperlipidemic Mice. Okay, the committee can reply.
Assalamualaikum warahmatullahi wabarakatuh. First of all, I will say thank you to the organizing committee of ISAC 2021 to allow me to present our results. The title of our research is The Effect of Nanoparticle Dewan Daru Fruit Extract to Reduce Total Cholesterol on Hyperlipidemic Mice. Our team are Onisa Triwalwafa, me, Marisa Angelina, Indah Dede Dewajanti, and also Visna Triana Dewi. The introduction of our research. We know that hyperlipidemia is a high-fat, cholesterol, triglyceride, or both level in the blood. Hyperlipidemia generally does not show specific clinical symptoms. The prevalence of dyslipidemia in Indonesia, based on total cholesterol concentration more than 200 milligram per deciliter, is 39.8%. Treatment with natural ingredient is an alternative to reduce the side effect caused by synthetic drugs because long period consumption drugs. In the study, we had been tested effect of the Dewan Daru fruit, Eugenia uniflora, as anti-hyperlipidemia. Eugenia uniflora is a plant that contains flavonoid, tannins, terpenoid, and essential oil, and had been used as traditional medicine, either in Indonesia and also in Asia. We know that it has specific fruit with attractive color. The active compound regarded of having potential as anti-hyperlipidemia are flavonoid, glycosides, and phenolics. The mechanism of action of this compound is to inhibit of the HMG reductase enzyme or inhibit cholesterol absorption. The figure shows us anthocyanin. Anthocyanin were reported has an activity as an antioxidants, anti-diabetics, and also anti-obesity. In research, we use chemical and instruments. First, Dewan Daru fruit that we obtained from Bandung, West Java, Sifasatin as positive control, 98% ethanol food grade, ketamine and selezins, cholesterol reagent from Elitex, natrium CMC, distilled water, and nanoparticle of Dewan-Daru extract that we got from our college. And also we use male mice who are aged 6 until 7 weeks with average of body weight plus minus 30 grams. We also prepare the extract. First, the pulp and fruit seed are separated and then drained in the oven at 50 degrees Celsius for 2 until 4 days. And then we macerate the dried fruit, use 98% food grade ethanol as a solvent. And maceration was carried out until the extract solution was colorless and the supernatant was evaporated, use rotary evaporator. In these experiments, we use several doses of Dewan-Daru, are 100, 200, and 400 milligram per kilogram body weight. And for nanoparticle of Dewan-Daru extract, the dose are 200 and 400 milligram per kilogram body weight. The dose of simvastatin as a positive control is 20 milligram per kilogram body weight. The animal that we use in this research is a DDY strain male mice. This study used a group of test animals, which consists of 4 animals in one group. The test animals were acclimatized for 7 days to adapt to the new environment by being given standard water and feed before starting the experiments. Hyperlipidemic feed for mice was made with the composition of 70% quail egg yolk, 10% fat, 20% sugar and butter. This feed was given for 28 days, and the extract and nanoparticle was given for 14 days after induction success. In this research, we calculated the serum, cholesterol concentration in serum. So the blood that was collected in a microtube, we have done centrifuge for 10 minutes at a speed 8,000 RPM, and then we separated the supernatant and was taken and stored at 20 and at minus 20 degrees Celsius until use. The cholesterol level of serum was measured using Elitex cholesterol kit. The 50 microliter serum mixed with the 500 cholesterol reagent, and then we do we homogenize and also we incubate it. And then we measure on the clinical spectrophotometry microlab. The data that we got, we analyze based on the percentage of reduction in total cholesterol level. We do the normality and homogeneity test before we calculate it using ANOVA, one-way ANOVA. The significant confidence level in this research, we use 98%. Sorry, 95%. The result of maceration, we get the rendezvous of extraction around 61.01%. The percentage of rendezvous quite high because we macerate the fruit, that we know the fruit rich of or high glucose and also high glycoside, so we can get many rendezvous. If we compare, then we extract a leaf. And for the data of cholesterol level, you can see the column that after feeding high cholesterol, we can see that induction use a hypercholesterolemia diet could induce cholesterol. This is normal control, give a normal fat, but from the number 2 until number 8, we give the hypercholesterolemia diet, and we calculated this data and use ANOVA. We can know that the data from number 2 until number significantly different with the normal control. It means our induction is success. Induction hypercholesterolemia is a critical point in the research because we cannot continue our study, give the treatment if the cholesteremia is not significantly between normal control and the treatment control. After then, we give the extract and also nanoparticle for 14 days, and we can see there are decreasing of cholesterol level in the serum. Actually, the normal control also show decrease the cholesterol and also negative control, but we calculated the percentage. From the percentage, we know that nanoparticle concentration to 200 milligram per kilogram body weight and also for 100 milligram per kilogram body weight, so the highest decreasing value. And we calculated this ANOVA one way. This result show that the decreasing significantly if we compare with the negative control. It's mean nanoparticle is potential to develop next to be a candidate of hyperlipidemic drugs. It's related with the theory that the ability of nanoparticle to increase the bioavailability of drugs with low solubility in systemic circulation has been widely demonstrated. In general, an increase in drug level in the blood is necessary for drug to cause pharmacological effect. We know that the Dewandaru fruit contain phenol and flavonoid, which have antioxidants, anti-inflammatory, and neuroprotective activities, while also being beneficial in glucose and lipid metabolism, so as to prevent obesity, diabetes type 2, and also dyslipidemia. Flavonoid act as antioxidant that can reduce lipid oxidation. Flavonoid cause inhibition of cholesterol synthesis, esterification synthesis of cellular cholesterol, triglyceride, phospholipid, and inhibits the activity of HMG-CoA reductase. So from our research, we conclude that Dewandaru fruit extract and nanoparticle form potential as a herbal for hyperlipidemia treatment, and the highest decrease or by nanoparticle Dewandaru fruit extract dose 205 and 400 milligram per kilogram body weight. We also thanks to Mr. Martoha Paratua Lumban Raja for giving Dewandaru fruit, Mr. Sukirno and Dedi Supriyadi for technical assistance. Thank you for all of your attentions. Assalamualaikum warahmatullahi wabarakatuh.
Waalaikumsalam warahmatullahi wabarakatuh. Yes, thank you very much for I think Bu Marisa. Thank you very much for the nice presentation. Okay, we're going to the next presenter. Will be presented by Nadira, but actually I will check the presence of Mrs. Nadira or someone who represent the Nadira is present here or not. Okay, I think it's not attending this meeting in this time. Maybe at the end of the presentation can hopefully can attending this meeting. Okay, we're going to the next presenter. Will be presented by Mrs. Yasmin Eka Pratiwi. She is a researcher from Research Chemistry with the title of its article, The Evaluation of Antibacterial Activity of Ultrasound-Assisted Hydroxylation Ammonium Carbon Oil. Okay, computer can play the video.
Good afternoon, ladies and gentlemen. My name is Yasmin Eka Pratiwi. Today, I would like to present our research titled Evaluation of Antibacterial Activity of Ultrasound-Assisted Hydrodistillation Ammonium Carbon Oil. Our team consists of Mrs. Professor Dr. Ani Sulaswati, Mrs. Risna Triana Dewi, Mr. Egi Agustian, Ms. Ade Rahmawati, and myself. This is the outline of our research. There are introduction, materials, methods consist of conventional and ultrasonic assisted hydrodistillation, gas chromatography analysis, antibacterial analysis, and statistical analysis. And there are also results and discussion, conclusion, and references. Cardamom is a spice made from the seeds of genus Eleutheria and Amomum plants. In Indonesia, there are two types of cardamom, but in this research, we use the species of Amomum cardamomum. Our main focus is the antibacterial activity, where it has been reported that it shows zone of bacterial inhibition ranging from 7 to 16 millimeter and minimum inhibition concentration ranging from below 2.34 to 18.75 milligram per milliliter against gram-positive and gram-negative bacteria. The composition of Amomum cardamomum oil are oxygenated monoterpenes, which represent 71.4%, and monoterpene hydrocarbons, which represent 36.9%. The extraction method affects the content and chemical composition of oil. One of the methods that we can use is the ultrasonic assisted hydrodistillation, or UAHD, because it results in more effective extraction, saving energy, solvent, and time consumption. The mechanism of ultrasound process is based on two main physical phenomena, which are diffusion and mechanical destruction. It has been reported that UAHD influenced the chemical composition of oil and its bioactivity. Therefore, reflecting on the potential of the Amomum cardamomum oil, the study was designed to evaluate its antibacterial activity extracted with conventional and ultrasound-assisted hydrodistillation. The cardamom that we used were collected from Sumatra, and the seeds obtained from peeling from the skin and crushed to obtain powder with 8 mass particle size. And for antibacterial activity analysis, we use these materials. The conventional hydrodistillation is conducted by submerging 100 grams of sample to 2 liters of water in a 3-liter round bottom flask equipped with Clevenger type apparatus for 6 hours. For ultrasonic assisted hydrodistillation, we use Q-Sonica sonicator using 30%, 60%, and 90% of power for 15 minutes as pre-treatment. Afterwards, the sample was processed with hydrodistillation. To analyze the chemical composition, we use gas chromatography analysis, and for antibacterial analysis, we assess on Staphylococcus aureus, Staphylococcus epidermidis, Escherichia coli, and Bacillus subtilis. And the methods we use are this diffusion technique for zone of inhibition, or ZOI, and microdilution assay for calculating minimum inhibition concentration, or MIC. Afterwards, the results of the antibacterial analysis was analyzed statistically using Kruskal-Wallis. And this is the results of our research. According to gas chromatography analysis, the major compound of Amomum cardamomum oil is 1,8-cineol, which covers up to 60% of total composition. The sonication enhances the cineol, the 1,8-cineol content even further, where the highest content is found in oil with 90% sonication power, containing 69.43% of 1,8-cineol. And this is the results of antibacterial analysis. We compare the activity based on the power that we use in sonication. The highest antibacterial activity was found on epidermidis, while the least antibacterial activity was found on E. coli. It means that gram-positive bacteria are found to be more sensitive to essential oils than gram-negative bacteria. It is correlated with the composition of the outer membrane of the bacteria. The gram-negative bacteria is rich in lipopolysaccharide, while for gram-positive bacteria, it is rich in unsaturated fatty acids. And this is the general antibacterial mechanism from essential oil. The essential oil break the bacterial cell wall, damage the cytoplasmic membrane and the membrane proteins. and this activity of essential oil depends on the composition of and volatile components. The results of antibacterial analysis was analyzed by Kruskal-Wallis analysis and showed that sonication power has no influence on the measurement of both ZOI and MIC. And this finding is supported by other studies where it has seen that sonication as pre-treatment in hydrodistillation does not affect the biological activity. The study also performed antibacterial activity of 1,8-cineol against aureus and epidermidis as a preliminary study with the concentration of 10 mg per milliliter and 100 mg per milliliter, but show no inhibition. This finding is similar to some reports where it reported inactivity of cineol. However, the minor components in Ammonium cardamomo are also known to be an active antimicrobial agent. But because the minor components only exist in a small percentage of total composition, there are many possibilities of the mechanism of Ammonium cardamomo oil. It is possible that the minor compounds have higher antimicrobial activity than major compound, or there is some type of synergism with the other active compound, or the combination of minor and major compound thereof were possibly responsible for the complete antibacterial activity. So this is the conclusion of our research. The ultrasonic assisted hydrodistillation generates more yield of EOs and 1,8-cineol as a major compound and shows similar antibacterial activity with EOs extracted with conventional hydrodistillation. This method can be implemented in industrial scale to yield more oil with relatively same bioactivity quality. And the last is the investigation of the most active antibacterial agent and its mechanism should be taken as consideration for the next step. So this is the end of our presentation. Thank you for your attention. Good afternoon, ladies and gentlemen. My name is Yasmin.
Okay, thank you very much for Yasmin for the nice presentation. Okay, we're going to the next presentation in this section. Will be presented by Bu Galuh. She is a researcher at the Research Center for Chemistry, BIN.
I am Galuh Widiarti, and today I will present our research about analysis of microbial contaminants of gambir extract. The title of our research is Microbial Contaminant Evaluation of Gambir, Karya Gambir Crops Extract is Antioxidant Agent for Pharmaceutical Use. The outline of our presentation consists of introduction, objective, materials and methods, results, and conclusion. The background of our research are free radicals pollute a lot the environment, contributing various chronic and degenerative diseases in humans. The problems can be reduced by radical scavenger compounds known as antioxidants. And everybody knows Indonesian medicinal plants are source of antioxidant agents. And one of the plants is gambir that has been used traditionally for treatment of burns, diarrhea, and sore throat in the Asian countries. And many use of or benefits of gambir extract for human health. For for example, as antioxidant and antidiabetic, antibacterial, antifungal, antiseptic, anti-cancer. And refer to the rules of Indonesia National Agency Drug and Food Control BPOM number present in the slide at 2017. that completed the BPOM number 12 at 2014 about how to make proper the bioactive compounds for herbs or drug raw materials should be minimized of any other impurities and bacterial contamination. And based on Indonesian National Standard of Gamir and Indonesian Herbal Pharmacopoeia 2 about quality requirement of traditional drug raw materials for Gamir. And the purpose of the research was to analyze the microbial contamination, including contamination by bacteria, yeast, and molds of Gamir extract after the extract being stored for 6 months as antioxidant agent. And the analysis of the microbial contamination will be analyzed by TBC and TFC assay, and the antioxidant activity of the extract will be analyzed by DPPH methods. The material of the research are Gamir extract that was produced by aqueous extraction process from the leaves and twists of Gamir plant that obtained from the traditional farmer of Bangka Belitung, and the reagent for antioxidant is DPPH and ascorbic acid as standard antioxidant, and for TPC, TFC test were nutrient agar, peptone dilution fluid (PDF), and potato destro agar. And the equipment of the research are climatic chamber for storing the extract, oven, incubator, UV viscosity spectrophotometer for analysis of the catechin and antioxidant activity of the extracts. The methods of the research first, the aqueous extraction process from dried leaf and twists of Gamir plant, and then the extract was stored in a climatic chamber for 6 months. And after that, the extract was analyzed the antioxidant activity using DPPH methods. The water and catechin contents of the extracts analyzed based on Indonesian National Standard of Gamir and Indonesian Herbal Pharmacopoeia 2 about quality requirement of traditional drug raw materials. And for TPC and TFC assay, according to Indonesian National Standard ISO, which number present in the slide, and the TPC and TFC of the extract were evaluation according to microbial contaminants evaluation referred to BPOM number 12 at 2014. The research of our study from the table, the research showed that no difference of water and catechin constraints from the fresh and extract was being stored for 6 months, and the water content of the fresh extract and extract was being stored for 6 months, approximately 14%. The catechin contents is also from the fresh and extract was being stored for 6 months were approximately 56%, and but the antioxidant activity of the extract based on activity value is strictly different from the fresh extract, and the extract was being stored for 6 months, where the fresh extract is little more active than extract was being stored for 6 months, and from the preliminary study and study literature, everybody knows that major compounds of the gamma extract is catechin. That catechin known as very active as an antioxidant. And based on Indonesia National Standard of Gamir, based on water and catechin contents of the extract, classified class 2 Gamir product, and if based on IC value that it are less than 50 microgram per milliliter, so that the extract is very active as antioxidant. However, its activity less active than ascorbic acid, with AC50 value is 10.97 microgram per milliliter. And from the microbial contaminant analysis from the table, the microbial contaminant analysis where the TPC and TFC were conducted triplo, and from the Petri 1, 2, and 3, both of the TPC and TFC, the TPC and TFC value showed that the TPC in TFC are less than 10 colony forming unit per gram. So based on Asian guidelines on limits of contaminants for traditional medicine and health supplements, that started TPC and TFC of plant extract materials for oral use using boiling water are not more than 5 multiple 100 million and colony forming unit per gram. So as well, the rules of the BPOM number with number present in the slide at 2017. So for the result of our research, we conclude that Gamir extract has strong antioxidant activity with IC50 value of 26.59 microgram per liter, and the extract with water and catechin contains approximately 14% and 60% respectively, so that the extract was categorized as class 2 of Gamir product, refer to the Indonesia National Standard of Gamir and Indonesian Herbal Pharmacopoeia 2 at 2017, and from the TPC and TFC values of the extract were less than 10 colony forming unit per gram. The extract is according with the quality required by the Indonesian Herbal Pharmacopoeia 2 and the BPOM regulation number 12 at 2014. This some reference of our research, and we would like thanks to Indonesian Ministry for Research and Technology for its financial support through INSINAS Act 2019, and as we also thank to Research Center for Chemistry BRIN that supports of our research, and we also thanks to Professor Muhammad Hanafi for his suggestion for this research. Thank you very much for your attention.
Thank you. Thank you very much for Bugalo for the nice and clear presentation. Well, we will go to the next session, but maybe I will check final call to Mrs. Nadira. Nadira is present here or still not? Yeah, okay, I think it's Nadira absent. Well, we will go to the next part. It's a discussion Q&A. And I have already read some question in the chat box. For the first one, the question from Mrs. Faiza Mariani Brin to Marisa. Yeah, I think Marisa have two questions. Yeah, maybe.
Okay. Can you hear my voice?
Yes.
Okay, thank you, Faiza, for the questions. I see in the list of questions there are two questions about nanoparticle. Actually, in the material method, I thought that we got nanoparticle from our colleagues, so it's not part of our research in this paper, because our colleague also will be published the method how to make nanoparticle of Dewan-Daru fruit extract. Is not part of this research. So I cannot explain more to Faiza about the how to make it and also how to prepare that. But for the question from Abdi, I continue to Abdi question. Is it okay, Eka?
Yeah, okay.
Okay. I continue to the question from Abdi. Yes, we can see the percentage of decrease value of cholesterol is nanoparticle of Dewan-Daru extracts more powerful compared with simvastatin. But we must also note it that dose is quite different. The dose of nanoparticle 10 times compared with simvastatin. Simvastatin 20 milligram per kilogram body weight, and nanoparticle 200 milligram per kilogram body weight and 400 milligram per kilogram body weight. Why we got the highest dose? Yeah, because we use like an extracts, not a pure compound. But simvastatin is a pure compound, and the usual dose is a 20 milligram. So the dose quite different. If we want to know, or we can know exactly about the how powerful nanoparticle, we must compare with the same dose maybe in the next experiments. But in these experiments, we cannot conclude that simvastatin below nanoparticle is more powerful because the dose is quite different. And how to prepare it, that I say before, that is our, we got the material from our colleagues, so we cannot explain more, but I little bit know that he just only homogenized, not use ball milling process, but only homogenized on high RPM, average 20,000. Sorry, I forgot about the method because we just only got from our colleagues. Thank you. And then the next question for me, I can continue to answer. Eka?
Yeah, I think until now only two questions. Yeah, maybe Mrs. Faiza or Mas Abdi will be a response or
okay.
Yeah. And maybe for the first one, Mrs. Faiza maybe. Yes.
Yes, I think it's all clear. That I know that you present that you got the sample from your colleague, but I just want to know the general method, whether the ball molding like Mr. Abdi said or something like that. But if it's not your, how to say, but if you could not tell more, so I think it's okay. Thank you.
You're welcome.
Yes, thank you. And maybe Mas Abdi will give the response on the answer.
Yeah, it's clear enough. Actually, I'm curious about the method how to prepare this nanoparticle, because since we know we have to how to say, we have to clarify that the scale, the particle size of nanoparticle. That's why I asked this question. Maybe Dr. Marisa already said that she got this sample from her college. That's great enough. Thank you so much.
Okay, thank you for your understand, Abdi and Faiza.
Thank you. Yes. Thank you. Maybe actually I have maybe little question for Bu Marisa.
Yes.
Yes.
One more question in the list.
Yes. Only two questions for Mas Abdi and Mrs. Faiza.
No.
Oh yeah.
Oh, sorry.
Oh,
No. There's question direct to me. So you cannot see, but I can see the question. Sorry. It's direct. Okay. Okay.
Yeah, I think in your research you already directly test to in vivo activity in the male list. Maybe why you do not try it on the in vitro activity maybe before you test the in vitro, and maybe is it available the methods to in vitro activity, the some kind of method to hypolipidemic yeah.
Yes, a nice question, Eka. Yeah, actually, to make more cheap and more either, actually, before we go to in vivo testing, first we do the in vitro testing. But in this case, especially in correlated with the cholesterol and like triglyceride or cholesterol, we are things that if we testing the in vitro use HMG CoA reductase enzymes is also quite expensive. Actually, if we cannot isolate from the liver of or hepar of mice, it's maybe more cheaper, but we cannot do the isolation of the enzyme. So we must buy the enzyme, which is the price is quite expensive. So for the cholesterol, we think better we continue directly to in vivo before and we didn't do the in vitro testing. But if next if we can isolate the HMG CoA reductase by our team, it's better to do the in vitro testing before we go to in vivo testings. And actually, this project, the purpose of this project is to report the result to submit to BPOM. BPOM in Indonesia didn't ask the in vitro result, but asking about the in vivo results. It's one of the reasons why we didn't do the in vitro testing. Okay, it's all.
Okay, yeah, I think it's clear. Yeah, thank you so much, Bu Marisa.
Yeah.
One more question can.
No, oh yeah, okay, please.
I just want to know about the simvastatin that you give your treat to your mice. You apply to your mice at night or morning, Dr. Marisa?
Yeah.
Because we know that in the clinical use, always simvastatin always taken at night, right? So in this study, in your animal study, you give the simvastatin at night or?
Yeah, actually, the simvastatin in patient eat in the night before sleep, but in the testing on animal studies, we give in the morning. But we know that the mice is different
design.
No.
Metabolism.
Different metabolism, and also in the morning until noon, the mice sleep. Different with us. We are sleep in the night, but mice active in the night and sleep in the morning until noon. Maybe it's one the reason why the testing on mice either in the clinical use at night, but in the mice testing give in the morning.
All right.
Yeah.
Yeah. Okay. Yeah, I think it's very interesting discussion. Thank you so much for Bu Marisa and Mas Abdi.
You're welcome.
Okay. We're going to the next questions. To Mrs. Yasmin. Yes, I think it's I have two questions. Maybe Mrs. Yasmin already read the question and time is yours to answer. Yes.
Okay. Thank you for the question, Mr. Abdi. We use ultrasonic because it results in higher yield and short time extraction compared to the conventional one. And from the several reports, it showed that the increasing of targeted or major compound, and in this case is 1,8-cineol. So our hypothesis, it will affect the bioactivity, or hopefully it will increase the bioactivity. But from the results, the ultrasonic didn't have significant influence on it. And for the next question, actually we use antibiotics as a positive control just in the preliminary study to make sure the bacteria suspension growth. But for the further assay, we don't use it anymore because we just want to compare the bioactivity from each oils. And I'm sorry, I don't present it here.
But because I see, I saw in your slide presentation, it looked like your essential oil have high MIC value, right, around 1.2 something milligram. That means 1,000 something. It looks not active at all, in my opinion.
Sorry, could you repeat?
I looked in your table, the MIC value of your antibacterial activity of essential oil around 1.2 milligram per ml, right? It means 1,000 something, 1,000 microgram per milliliter. I think it's not, how to say, it's not be recommended to say that it's have antibacterial activities, because if we apply it directly to animal or in vivo, it needs high amounts of essential oil to kill bacteria. So I think, and all that's why you have to, and also in your slide you didn't show the positive control. So I'm confused whether the concentration it's look jump something like that.
Yeah, actually it is related to the question from Mr. Ahmad Darmawan. So maybe I can answer this question and also discuss with you. And yeah, for this moment, we can say that 1,8-cineol show poor antibacterial activity shown from our research and also supported by other findings. But however, we cannot conclude that the cardamomo essential oil has poor antibacterial activity because we didn't know yet what compound as a key player which has the highest antibacterial activity. So we need further studies to investigate it.
Yes, that's the answer. So maybe Mr. Ahmad, maybe we'll have the response for the answer.
Okay, clear, Mas Eka, clear. Thank you.
Yes, thank you. Yes, thank you. Yeah, I think the study from the Yasmin and team. Yeah, I think the suggestion for Mas Abdi is very important. The positive control, right? Positive control is important to testing the antibacterial activity, and I think other BIC and also use positive control. But for all, I think very nice and interesting project.
Okay,
thank you.
Thank you
Mr. Yamin,
for the advice, Mr. Abdi and Mr. Ahmad Darmawan.
Okay, yes. Okay, we're going to the next questions. Well, yeah, I think it's the special questions. Yeah, because this question regarding to the presenter who present the first sessions. I think it's no problem, right? No problem, because Mrs. Jatri is still in this room, and I think it's available to answer the question. For the first one, question from host to Z3. What is the different effect between sonication and conventional stirring on the products? Okay, Mrs. Z3 can answer the questions.
Thank you very much, Mr. Reka. And I would like to try to answer questions, even though my session is already finished. And actually, the two questions is almost similar. First from Mr. Asa, and the second one is from Ms. Yani. Both of them asking about comparisons of the product, if I'm not mistaken, whether using the conventional stirring method or with the ultrasonicator will be different or not. In visualizations, actually, the cinnamic acid of 4-methoxy with the 4-methoxyl groups in cinnamic acid is not soluble in ethanol. Impressively, even though we just used the sonication, which is only giving some typically fibrations into the solutions, the reactions can disperse or to all of the representative ester can easily to convert into the acid form, even though only using the fibration that occur from the sonicator. And from the based on the NMR studies, the two methods didn't give any different structural differences between the conventional and also the sonication method. And additionally to Ms. Yani, the question is whether should I use the sonicator or ultrasonicator. Actually, my research is not focusing on the size of the product, but the success of the conversions from the ester form to its hydrolysis form. Therefore, for in our the aim of our research, in my opinion, we only sonicate only the I mean the normal or the usual sonicator is already sufficient to convert the reaction. Is that clear for my answer? I would like to call for the moderator.
Yes, I think it's clear answer, but from co-host, Bayani maybe will have some response regarding the answer. Okay, I think it's clear. Yeah.
Thank you very much.
Yeah, but I'm sorry, but continuing the question, I remember that you mentioned that your research, your study, focusing to developing the particular methods, right? Not the product. Not the focusing to how to develop the particular method to obtain some kind of the pure compound. And my question is, what the different between your method and the previous method, and what the advantage of your methods that you have been established?
Actually, thank you very much for Mr. Eka. Actually, the product is less soluble in ethanol. Therefore, I think using sonication, which is how to say, there are no conventional stirring method used in the typical reactions. Therefore, in my opinion, only use the giving the fibrate into the reaction can trigger the reaction. So, how to say, by minimizing the kinetics of the reaction can also result into the excellent product of the hydrolysis product. So compare with the previous method, actually none of them are utilizing the sonication for this type of reaction, especially in cinnamic acid.
Okay, thank you. I think it's clear. Yeah, thank you very much.
May I have one suggestion to Yasmin? That's,
okay, please. Yes.
Because I see you got essential oil in this research and have how to say, we can say that's poor activity against bacteria. So I mean, I think you can use another microorganism to test it. Your essential oil, for example, you use fungi. Maybe your essential oil have the activity for the dermatophyte, dermatitis, something like that. So maybe your essential oil cannot kill bacteria, but have activity against fungi, something like that. Because fungi and bacteria have different mechanism of actions. So I suggest you, if you want to know, if you want to focus on the what we call it for topical application or something like that, you don't see the activity on bacteria, you can move to another microorganism that's that cause the dermatitis. You can use Ticopiton rubrum or something. So just straight away. So just move on. Let do your research. Don't move to another microorganism. So don't focus on bacteria.
Okay, thank you for the advice, Mr. Abdi. It means a lot.
Okay, yes, thank you. Yeah, thank you very much for Ms. Abdi and for the nice suggestion. Well, actually, maybe some participants already still have questions to the presenter. We will have 5 minutes before we end this session. If not, maybe for the last question, I have a question to Bu Galuh. Yes. Yes, Bu Galuh. Actually, your research is focusing to check. I think I focus to microbial contaminants. Yes. In your methods, you check the viability of the microbial, including bacteria or fungi. I think I see that only check to grow in the medium. I think it's common medium, nutrient agar and potato dextrose. And my question is, it is enough to make sure that no bacterial contaminant, because I think some bacteria or fungi will grow needing the special condition on special media, not only in an nutrient agar or a potato dextrose agar. So yes, I think I would like to give the response regarding that issue. Right.
Thank you, Mas Eka. Yes. In my study, we also analyze the RO water as the solution for the solution of the product, and we also the analysis of the TPC and TFC of the media. And I'm sorry, but the result of the TPC and TFC of the media and the RO water, we not present on this. Thank you.
Oh, okay. Okay, thank you very much, Mr. Kal, for your confirmation. I think yes, I think this is the end of this session. Yes, actually, I would say thank you very much for the interesting and warm discussion during this section. And for the announcement, maybe after I close this meeting, please come back to this room at 15 and 50 p.m. or 15 minutes after we closing this meeting. Okay, that's all of my of this session, and thank you very much again for your participation, and thank you. Assalamualaikum warahmatullahi wabarakatuh.
Waalaikumsalam warahmatullahi wabarakatuh.
Is one of the most dynamic and innovative companies within the Berke Group. The Berke Group consists of over 35 operating companies in over many industries, including technology, infrastructure, and engineering. Berke America has been in business for over 17 years and has established itself as the major player in the research and the chemical analysis industries. Its customers include many bodies in the government sector, as well as large corporate entities. And its industries include food research, agriculture, petroleum, chemical, environment, and clinical research. We believe that Berke America is very well suited to meet with all stakeholders to support the quality of life and to improve it in Indonesia. We fully support Berke America to meet with all stakeholders and form partnerships to improve the quality of life of people in Indonesia. Is one of the most dynamic and innovative companies within the Berke Group. The Berke Group consists of over 35 operating companies in over many industries, including technology, infrastructure, and engineering. Berke has been in business for over 17 years and has established itself as the major player in the research and the chemical analysis industries. Its customers include many bodies in the government sector, as well as large corporate entities. And its industries include food research, agriculture, petroleum, chemical, environment, and clinical research. We believe that Berke is very well suited to meet with all stakeholders to support the quality of life and to improve it in Indonesia. We fully support Berke to meet with all stakeholders and form partnerships to improve the quality of life of people in Indonesia.
is one of the most dynamic and innovative companies within the Berke Group. The Berke Group consists of over 35 operating companies in over many industries, including technology, infrastructure, and engineering. Berke has been in business for over 17 years and has established itself as the major player in the research and the chemical analysis industries. Its customers include many bodies in the government sector, as well as large corporate entities. And its industries include food research, agriculture, petroleum, chemical, environment, and clinical research. We believe that Werken Niaga Medica is very well suited to meet with all stakeholders to support the quality of life and to improve it in Indonesia. We fully support Werken Niaga Medica to meet with all stakeholders and form partnerships to improve the quality of life of people in Indonesia. is one of the most dynamic and innovative companies within the Berke Group. The Berke Group consists of over 35 operating companies in over many industries, including technology, infrastructure, and engineering. Werken Niaga Medica has been in business for over 17 years and has established itself as the major player in the research and the chemical analysis industries. Its customers include many bodies in the government sector, as well as large corporate entities. And its industries include food research, agriculture, petroleum, chemical, environment, and clinical research. We believe that Werken Niaga Medica is very well suited to meet with all stakeholders to support the quality of life and to improve it in Indonesia.
We fully support Berkepribadian Medica to meet with all stakeholders and form partnerships to improve the quality of life of people in Indonesia. is one of the most dynamic and innovative companies within the Berke Group. The Berke Group consists of over 35 operating companies in over many industries, including technology, infrastructure, and engineering. Berke has been in business for over 17 years and has established itself as the major player in the research and the chemical analysis industries. Its customers include many bodies in the government sector, as well as large corporate entities. And its industries include food research, agriculture, petroleum, chemical, environment, and clinical research. We believe that Berke is very well suited to meet with all stakeholders to support the quality of life
Mbak Sus, can we start now? Okay. Bismillahirrahmanirrahim. The speakers, distinguished guests, ladies and gentlemen, good afternoon. Assalamualaikum warahmatullahi wabarakatuh.
Waalaikumsalam warahmatullahi wabarakatuh.
First of all, I would like to welcome you all to this prestigious seminar, the fifth International Symposium of Applied Chemistry Webinar Conference, especially in the field of natural product and medicinal chemistry, pharmaceutical technology, and drug delivery system topics. In this occasion, we shall have outstanding presentation from our prominent presenter, whom I believe shall enrich our insight and knowledge regarding the theme of this seminar. I am Sofa Fajriah, will be as your moderator on this session. Before we begin this session, I will check the presenters first. May I absent for Miss Kartika Diah-Palupi is here?
Yes, I'm already here.
Okay, thank you. And next is Miss Eni Soliha.
Yes, Miss Sofa.
Okay, thank you. And Mr. Yusuf Andriana is here. Hello, Mr. Yusuf Andriana, are you in here? Okay, maybe Mr. Ahmad Darmawan. Okay.
Okay, here.
Yeah. And Mr. Ade Chandra Iwansyah.
I'm here, Miss Sofa.
Okay, thank you. So we start this session. We have 5 presenters in this session. Each presenter has 10 minutes to present their presentations, and Q&A will be doing after the fifth speaker's presentation. And as your information, you can raise your hand or chat or unmute your mic to ask your questions. Okay. Our first presenter is Ms. Kartika Diah-Palupi. Kartika Diah-Palupi. She will deliver a presentation with entitled "Efficacy of 2-2-axane epitoskirin A in a mouse model Staphylococcus skin abscess. Yeah. Okay, ladies and gentlemen, please welcome Ms. Kartika and please the co-host to play the video. Thanks.
Hello, everyone. I hope all of you have a great day today. And first of all, I would like to thank the ISEC 2021 committee for giving me the opportunity to present our research. And today I will present our work entitled Efficacy of 2-2-epitoskirin A in a mouse model of Staphylococcus aureus skin abscess. This was a collaborative work between Dewi Lansari and myself, Kartika Diah-Palupi, and also Pratiwi Arif Nurtanto as well as Andrea Agusta. And we are all from the National Research and Innovation Agency, or PRIN. So I'll begin with the background of this study. So in our laboratory, we have successfully isolated a promising novel antibacterial agent called 2-2-epitoskirin A, or we called it EPI-A. So this is the EPI-A here. EPI-A is a bisanthraquinone produced by an endophytic fungus, Diaforte species GNBP10. We isolated the endophytic fungus from a plant called Funcaria gambir. And in the previous in vitro studies, EPI-A displayed excellent activities against some bacterial and fungal strains, notably against S. aureus, with MIC value of 0.06 microgram per milliliter. And as we all know, S. aureus is bacteria that causes various human diseases, including meningitis, endocarditis, pleuropulmonary, as well as skin and soft tissue infection. And for the skin and soft tissue infection, an abscess is one of the most frequent manifestations. This is because S. aureus produces virulent factors and bacterial molecules such as lipoproteins that promote the formation of skin abscesses. So from the previous in vitro study, we would like to further our investigation And in this study, we would like to evaluate the efficacy of 2,2-epistoschirin A against S. aureus in an in vivo system. So we use a DDY mouse model of S. aureus gene abscess. As for the method, first we induce neutropenic condition in the male DDY mice by injecting cyclophosphamide 4 days and also 1 day before the infection. We then randomly group the mice into treatment groups, which is the epistoschirin 12.5, 25, 50, 100, and also 200 milligram per kilogram body weight, and also control groups, including the 2 groups of positive control, which were vancomycin 4 and 16 milligram per kilogram body weight. And also 1 group of carboxymethylcellulose sodium 1%, which was the vehicle for the drug, and we used it as untreated control. And 1 group of active control where we injected the saline solution instead of the S. aureus suspension. And then for the treatment and infection, 4 hours before the infection, the control and epi groups were administered orally with corresponding substances, except for the vancomycin group, because the vancomycin was administered subcutaneously 1 hour before the infection. And then we proceeded to the infection, and for the infection, we injected 0.1 milliliter suspension of 1 million CFU per milliliter clinical isolate of S. aureus subcutaneously into the abdomen of previously shaved male mice. And then we observed and measured the abscess daily. And at day 14, the animal were sacrificed, the abscess were excised, weighted, and also homogenized. And then we made serial dilution of abscess slurries to count the number of viable cells. And then also we performed statistical analysis to compare the treatment and also the control group. So this is the result. And as we can see here, that at the higher dose of epistoschirin A, the abscess area were bigger rather than smaller. And at the dose of 12.5 milligram per kilogram body weight of epistoschirin A, the abscess area was smaller than the higher doses and comparable to the vancomycin groups, which were the positive control. And as you can see here, the abscess area of epistoschirin A 12.5 milligram per kilogram body weight was significantly smaller than the untreated control. And then for the bacterial count, it also showed similar results with the abscess area at the dose of 12.5 milligram per kilogram body weight. The count was significantly lower than the untreated group. So as you can see here, that the healing effect of 2,2-epistoschirin A was only observed in lower dose and tended to get worse at higher doses. We assume that this is because the immune system might be disturbed in higher doses, and as the consequences, the abscess area were getting worse. For a previous study, we know that even though EPA is only slightly toxic with LD50 around 1,600 milligram per kilogram body weight, an in vitro study in a macrophage cell line showed that at the concentration only 1 micromolar of epistoschirin A, it inhibited 55% of the cell growth. But at low doses, like for example 0.01 micromolar and then 0.1 micromolar, it only inhibited 4 until 7% of the cell growth. So this is interesting because, as we all know, the macrophage cells play a pivotal role in the immune system to combat infection. However, in this study, the immune cells of the mice itself was not monitored too. The state of the animal immune system among the groups could not be compared. So for the conclusion of this study, first is that 2,2-epistoschirin A effectively inhibit abscess area and number of S. aureus bacterial count in the dose of 12.5 milligram per kilogram body weight orally. And then the effectiveness of the substance is less as the dose increases. Since the dose of 12.5 milligram per kilogram body weight is the lowest dose in the study, there is a possibility of doses lower than 12.5 milligram per kilogram being effective in inhibiting the incidence of abscess. And then the unusual response observed in a higher dose has led to the presumption of other activities that might be possessed by 2,2-testosterone A, especially in immune system suppression. However, we need further study to confirm the statement. Okay, that's all. This is the end of my presentation. Thank you for your kind attention. Hello.
Thank you very much, Ms. Kartika, for a very nice presentation. Well, now, ladies and gentlemen, let's welcome our second presenter. She will deliver a presentation with entitled Isolation and Characterization of Catechin from Cascara as a Potential Source of Antioxidant and Antibacterial. Ladies and gentlemen, please welcome Ms. Annie, and please the co-host to play the video. Thanks.
Assalamualaikum warahmatullahi wabarakatuh. Good afternoon. I am Annie Salihah. I'm from Research Center for Appropriate Technology, National Research and Innovation Agency. Now I will talk about purification and characterization of catechin from cascara and its evaluation on antioxidant and antibacterial activities. Coffee is the most popular beverage in the world. There are many species, almost 120 species, but the most important or the most popular in coffee industry are Arabica and Robusta coffee. In coffee production process, we also get the coffee hash as byproduct. It is about 40 until 45%. And the coffee hash is raw material of cascara. Several studies reported that cascara is a potential source of antioxidant because it contains phenolic compound, and according to Ramirez Martinez, the most abundant phenolic compound in the coffee cherry pulp are chlorogenic acid and epicatechin or catechin compound. So the study about active compound in the cascara will provide information about the benefit of consuming cascara. In our research, we isolate and characterize the catechin from cascara, Robusta, and Arabica, and also we test the antioxidant activity and antibacterial activity. Okay, from the method, first is we prepare the cascara from coffee cherry, and then we sorted the cherry to remove the impurities and the overripe cherry, and then we wash and pulp the cherry to separate the coffee bean and coffee hash, and then we dry the coffee hash at 45 degrees Celsius, and then we chop to get the cascara. The next step is isolation of catechin from cascara. It was about 20 grams of cascara. We measured it with ethanol, 95% for 24 hours. And then we evaporate the ethanol or the solvent. And then we fractionate the extract using column chromatography with acetone water in ratio 1:1 as mobile phase and silica gel as the stationary phase. After we have isolates from cascara, Arabica, and Robusta cascara, we characterize the isolate using thin layer chromatography, UV visible spectrophotometer, and FTIR spectrophotometer. In this characterization, we also use catechin standard to verify or to compare the result. And then we test the antioxidant and antibacterial activity. DDPH method was used for antioxidant activity, and the value of antioxidant activity is on the RSA. RSA is radical scavenging activity that we calculate based on this equation. And then the antibacterial activity was tested by a distribution method. Okay, this is the result and discussion. Table 1 is about the yield of cascara isolate. For Arabica cascara, we have 6 fraction of 6 isolate and 5 isolate for Robusta cascara. And then we characterize the isolate first using UV visible spectrophotometer to identify the wavelengths, maximum wavelengths, and we can see that this is, it is different between the isolate and the catechin standard. The different value because of the other component in the isolate, such as caffeine and its derivative, like the result of SQL and Geminis. And according to Sari and Kuntari, caffeine has wavelengths at 273 nanometer. And from the table 2, we can see the fraction number 3. It has 2 peaks that indicate the glycoside compound. So we can conclude that the isolate not only contain catechin but also other component like caffeine and glycoside. This is the FTIR spectra of the fraction. We can see from this spectra that this is similar, but between catechin standard and the Arabica fraction and Robusta fraction. In this wider peak is very characteristic for hydroxyl group, and then this is for the ring of vinyl, and then in the peak in the 1095 until 1018, it is for COC ether. And this is the difference between the catechin and the fraction. It is indicate the CH stretching. Okay, the next is antioxidant activity. Table 3 show the antioxidant activity of quercetin and also the fraction Arabica and also the Robusta. You can see that the quercetin has IC50 less than 50 ppm. It's about 25 ppm, so we can classify the quercetin is a very strong antioxidant. But the isolate, we have strong antioxidant characteristic because in the IC50 is about 50 ppm, except for the fraction number 3 for Arabica and fraction number 1 for Robusta. This is not the lowest antioxidant activity. In the right side, you can see the structure of catechin and quercetin. They have a hydroxyl group that can stabilize the radical from DPPH. This is the scheme of the reaction between the DPPH and antioxidant compound. Okay, the last is about antibacterial activity. In table 4, we can see the inhibition zone of the fraction for Escherichia coli and Staphylococcus aureus. And according to Davis and Sod, that the inhibition, the classification of antibacterial activity is on the inhibition zone diameter. This is the classification. And this fraction, we can see that this fraction had moderate to strong antibacterial activity against 2 types of bacteria tested. Thus, the diameter of the inhibition zone
Mbak Sus, can you hear me? Is there any technical?
Yeah, wait a minute, Abu. Wait a minute.
Okay, thank you.
But I think Ms. Sofa maybe just only one slide again for conclusion.
Okay. Still waiting the video. Maybe I just call Mr. Yusuf Andriana. I see in the participant, Mr. Yusuf Andriana is present in here. Are you hear me? Mr. Yusuf Andrian?
Mr. Yusuf.
Yes, I'm here.
Okay, thank you. After Ms. Solehah, yeah? Okay.
And then for the diameter of the inhibition zone of Arabica and Robusta sample against Escherichia coli was in the range of 8 until 12. So the antibacterial activity of Arabica and Robusta against this bacteria was classified as moderate until strong. For conclusion, the first isolates of catechin compound from Arabica and Robusta cascara were obtained by maceration with ethanol 95% for 24 hours and column chromatography with acetone water as mobile phase and silica gel as the stationary phase. And the second, the yield of catechin isolates from cascara arabica was 0.02% and for Robusta was 0.01%. And the third, the catechin isolate of Arabica cascara at the concentration 50 ppm had antioxidant activity 78% and for Robusta 76%. And the last, catechin isolate of Arabica and Robusta had moderate to strong antibacterial activity against Staphylococcus aureus and Escherichia coli. Okay, thank you. Assalamualaikum warahmatullahi wabarakatuh.
Waalaikumsalam warahmatullahi wabarakatuh. Thank you very much for a very nice presentation. Okay, now we're going to the next speaker, the third speaker. Mr. Yusuf Andriana will deliver a presentation with the entitled an overview of allochemicals and allopathic activity of Alpinia malacensis. Okay, ladies and gentlemen, please welcome Mr. Yusuf Andriana, and please the co-host to play the video. Thank you.
Thank you for the opportunity that has been given to me. My name is Cecep Erwan Andriansa from BRIN. I represent fellow researcher consists of Yusuf Andriana, Ade Chandra, Asri, Cahya Edi Anggara, Wahyu Anggara, Diki, Rohma, Hendarwin, Nur Katika, Nur Haidar, Christina, and Truong. I want to present the result of our research. The title is an overview of allelochemicals and allelopathic activity of Alpinia malacensis. Introduction. Plant pathogen and weed are the major contributor to reduce crop yield and product quality. Is an agricultural system by competing the crop for resources like nutrient and light or attacking directly on plant tissue and cold case yields loss for more than 50%. On the other hand, the overuse of synthetic pesticides or herbicides has negative effect on organisms around crop field and cause environmental damage. Allelopathic approach is an alternative solution that utilizes allelochemicals from plant or microorganisms to control pathogens and reduce environment risk. Allelopathy is defined as the interaction among a plant or microorganism is the ecosystem by chemical exudation into the environment. The chemical is called allelochemicals and can be utilized to control weeds or plant pathogens. Several studies claim that this approach is safer and gives low risk and environmentally effect. Alpinia malacensis is a medical plant that has been widely used to treat several diseases such as nausea, vomiting, and certain wounds, and also a seasoning ingredient in processed meat. It belongs to Zingiberaceae family and widely distributed in tropical region of Asia, including Southeast Asia, Indochina, Bangladesh, and Sri Lanka. Searching method from Scopus, Web of Sciences database, of Sign of databases, and Google Scholar search for articles were conducted using the keyword Alpinia malacensis, allelopathy, and Alpinia malacensis, antioxidant and Alpinia malacensis, fungicidal and Alpinia malacensis, herbicidal and Alpinia malacensis. The publication till May 2021 were then classified and analyzed to biological activities related to allelopathy and the allelochemicals. After screen, 45 literatures found in scientific databases will possess 20 literatures that relevant to allelopathic activity of A. malacensis. Allelopathic and relevant biology activities of malacensis. In the table 1, rhizome of A. malacensis showed a higher antioxidant activity that is lead but lower than its essential oil. Similarly, in total phenolic content, essential oil part of A. malacensis showed the highest value, 26.59 milligrams gallic acid equivalent per gram. Then rhizome and leaf extract. It seems essential oil from malacensis possesses more active component than the rhizome and leaf against DPPH and ABPS. Antibacterial activity of malacensis. Antibacterial activities of malacensis from several studies are illustrated in table 2. In rhizome extract, hexane extract showed more inhibitory effect on L-monocytogenes than ethanol extract. In contrast, ethanol extract exerted more suppression on E. coli. Essential oil of E. malacensis showed inhibitory effect on the growth of both gram-positive and negative bacteria. Pseudomonas aeruginosa seems the most sensitive against essential oil of malacensis, indicated by the smallest value of IC50, 13.667 milligram per milliliter. For isolated compound, caprolactone showed a higher inhibitory effect against some bacteria than caprolactone. Antifungal and herbicidal activities of malacensis. Fungicidal activity of malacensis extract or essential oil is illustrated in table 3. Essential oil from this plant shows more potent inhibitory activity against R. solani, S. sclerotinia, and S. rolfsi. Compared to the other extracts, methanol possesses are stronger inhibitory activity against than hexane and dichloromethan extract. Herbicidal activity of amylase has been reported against Meremia peltate, and solid germination inhibition was 38.89 and 36.11% for rhizome and leaf extract at the dose. Allochemical of amylase is solid in Table 4. Allochemical from amylase are varied and consisted of different class of compounds, of which terpenes were identified as the major component extracted its essential oil. The other compounds, such as phenolic, flavonoid, and pyrrones, were also detected as allochemical from amylase. Almost of allochemical from amylase show with potent antibacterial activity. Conclusion: Alpinia malacensis is a medicinal plant possesses numerous potential of active component that gives advantages for pharmacological and allopathic activities. The important phytochemical from rhizome of amylacensis are cavalactone, palacavalactone, 5,6-dehydrocoine, coronarin E, coronarin A, naringin, quinic acid, and catechol. In the other hand, methylcinnamide, alpha-pelandrene, and 1-acetyl content in essential oil of 8-malacensis show potent beneficial allopathic properties. The study shows many beneficial allopathic activity of malacensis, particularly in antioxidant, antibacterial, fungicidal, and herbicidal activities. The presence of allochemicals such as terpenes, phenols, flavonoids, and pyrrones might be related to the allopathic activity of malacensis. Future research. Although in these studies describe many beneficial allopathic properties of malacensis, the study on allopathic assay against some plant indicator or which is still rarely found in scientific database. Therefore, the future research can be conducted in a malacensis study or germination and growth inhibition against several plant indicators. Determination of inhibition mechanism, each allochemical and application in paddy field also important aspect that can be further study to improve agricultural crops. Thank you.
Thank you very much for very nice presentation about the Alpinia malacensis. Okay, now going to the next speaker, the first presenter, Mr. Ahmad Darmawan will deliver a presentation with entitled Isolation and Identification of Beta-Testosterone Compounds Isolated from Makarana Natural. Please welcome Bapak Ahmad Darmawan and please the co-host to play the video. Thank you.
Thank you for the time and opportunity. Today, I would like to present part of our continuous project about drug discovery with the title Isolation and Identification of Beta-Testosterone Compound Isolated from Makarana Natural. This is one of our collaboration research between Research Center for Chemistry, BRIN, with Department of Chemistry, Faculty of Mathematics and Science, Halu Oleo University. As we know, Indonesian population is quite high and it direct correlated with health problems such as degenerative diseases like diabetes, cancer, and so on. Usually, we treat the disease using modern drug or chemical drug. But in the other side, Indonesia also known as one of the mega biodiversity country, which means Indonesia is rich with natural product resources, including plant resource. Unfortunately, until today, only few of them have been explored and utilized, and these numbers are not equal if we compare with the rate number of forest destruction. With the two conditions mentioned above, let us to the question about, is there any natural product resources, especially plant resources, can be used to substitute modern drug to treat some disease? Based on that question, we start to do drug discovery research based on local plant or endemic plant resources. And for today, I will present about phytochemical study of one local plant resource, Macaranga magnaturil. Macaranga, locally known as Mahang or Mara, ecologically identified as one of the pioneer plants with more than 300 species. Previous phytochemical studies saw that this genus is sources for alkaloid, flavonoid, tannin, stilbenoid, and steroid compound. More than 190 secondary metabolites have been isolated from this genus, and this genus also have various biological activities such as anti-cancer, anti-inflammatory, antioxidant, antimicrobial, anti-diabetic, and so on. But there are lack of publication about this species. That's why we choose this species to be explored. At the first, our sample collected from Mekonga Forest was identified at Herbarium Bogoriense Research Center for Biology. And our laboratory work start with sample maturation process using methanol, and the methanol extract further evaporated to obtain methanol crude extract. Methanol crude extract further fractionated with N-hexane, ethyl acetate, acetone, and butanol. Acetone fraction gave the highest yield of fraction compared to another fraction. And this acetone fraction further subjected to silica gel column chromatography. In this process, gradient polarity solvent system consists of N-hexane and ethyl acetate were used as eluent, and 22 fractions were obtained from this step. All the fractions were taken their TLC profiling, and from the TLC research, we will see that subtraction 11 saw an interest spot with intense light blue spot under UV lamp. We can see in the picture, based on the TLC result, the subtraction 11 further subjected to another silica gel column chromatography, and recrystallization and purification process of subtraction 11 led to the isolation of compound 1 and compound 2. Compound 1 and compound 2 further characterization using serial spectroscopic instruments such as NMR, proton NMR, carbon-13 NMR, HMQC, and HMBC. Also using LCMS or GCMS to find the mass. That you all can see in this slide, the NMR and LCMS data from compound 1 show the same profile and chemical shape of NMR spectra and also for mass spectra value with our first publication. But in our first publication, the compound obtained from ethyl acetate fraction. So for the compound 1, we are sure that compound 1 is beta-sitosterol. For compound 2, proton NMR show that this compound have 6 methyl groups and 1 olefinic proton. This proton profile of 6 methyl groups and 1 olefinic proton, you can see in proton NMR spectra, is characteristic for beta-sitosterol compound. From the carbon-13 spectra, we can also find that this compound have 6 methyl carbons, 2 olefinic carbons, and 1 carbonyl group. With the total carbon amount is 29 carbon atoms. After collected data from proton and carbon NMR, we also collect data from the HMQC and HMBC spectra. These spectra were important to find or to see the correlation between proton and carbon. And this spectra also an important data for postulating proteins to determine the chemical structure. From the correlation data we got, we can elucidate the data become the chemical structure like we can see in this slide. The correlation arrow in this chemical structure were generated from HMQC and HMBC data, where the blue arrow saw a direct H2C correlation or what we know as HMQC correlation, and the other black arrows saw indirect proton to carbon correlation or what we know as HMBC correlation. The chemical structure postulate should in accordance with mass value of the compound. And for compound 2, the mass value of 412. And this value is suitable for molecular formula of 29C, 48H, and O. From proton NMR data, C13 data, HMQC data, HMBC data, postulate structure, and also molecular weight data of compound 1, support with comparison data of beta-cytostenone from appropriate reference. In this case, Tang et al. All the data we got, including postulate result and comparison data, result indicated that compound 2 is beta-cytostenone compound. This is the last slide for the conclusion. We can conclude that 2 compounds we were successfully isolated from the lip of Makaranga material is beta-sterol compound groups, which are compound 1 is beta-cytosterol and compound 2 is beta-cytostenone. This is the first report for isolation of beta-cytostenone compound from Makaranga material. Thank you for your kindness.
Thank you, Mr. Ahmad Darman, for very nice presentation regarding the isolation and identification of beta-cytostenone compounds isolated from Makaranga material. Now we're going to the last speaker. The last speaker is Mr. Ade Chandra Iwansha. He will be delivered about the relationship antioxidant properties, phytochemical and toxicity profile of wild adalah, Choix lacrem jobivalent agrotis extracts, a principal component analysis approach. Please, for the co-host to play the video. Thank you.
Bismillahirrahmanirrahim. Assalamualaikum warahmatullahi wabarakatuh. Thank you, moderator, for this opportunity. Let me introduce myself. My name is Ade Chandra Iwansha. I am a senior researcher at the Research Division for Natural Product Technology BRI. In the International Symposium on Applied Science 2021, we will present our research report with entitled Relationship Antioxidant Properties, Phytochemical and Toxicity Profile of Wild adalah Extract, a Principal Component Analysis Approach. Next, for introduction. Chronic diseases such as cancer, diabetes, and cardiovascular could be prevented by the consumption of the whole grain. Several research revealed that the phytochemical in the whole grain have a role as antioxidant source and be able to reduce free radicals that dangerous to human body. Phytochemicals such as phenolics, carotenoid, and alkaloid as a bioactive compound are commonly obtained in whole grain, vegetable, herb, and fruit. Adalah, or Choix lacrem jobivalent seed can be used to make a variety of products such as cereal, fodder, and necklaces. In several countries, adalah has been widely used as a medical plant. In China, several traditional medicine usually use adalah to treat rheumatic, anti-hemolytic, and anti-inflammatory agent. However, the study did not investigate the relationship with the toxicity profile and did not use some statistical software approach. Several studies for adalah also had a role as a probiotic, treat diarrhea, and prevent colon cancer. However, only a few studies report the relationship of phytochemical profile, antioxidant activity, and toxicity of adalah. So, you can see that the objective of this study was carried out to evaluate the relationship, antioxidant properties, phytochemical, and toxicity profile of different various extract of wild adalah variety agrotis. The principal component analyzed